And we'll move on to the next speaker, which is Llorenç Cabrera-Bosquet from INRA in Montpellier, and he will talk about high-throughput estimation of incident light interception and radiation use efficiency of thousands of plants in a phenotyping platform.
Thanks, Bettina, and to the organization for choosing our talk. So I'm going to talk about the methods that we have developed in the PhenoArch phenotyping platform for estimating incident light interception, and radiation use efficiency in thousands of plants. So as a context, understanding the controls of biomass accumulation is a major challenge in the context of climate change. According to the question of Monteith, we know that biomass can be expressed as a function of the incident light reaching the crop, the amount of this light that is intercepted, that is basically depends on the leaf area and the architecture of this crop, and finally, and how efficiently this intercepted light is converted to a biomass, the radiation use efficiency that depends basically on photosynthesis. So, taking advantage of the possibilities of phenotyping platforms, we have developed a suite of methods for deriving each one of the terms of this equation. And we tested whether these radiation use efficiency values were stable between experiments and related to leaf gas exchange. And finally, we extend these methods for extracting other related traits to light interception, like interception efficiency, and like the extinction coefficients from virtual experiments. So just briefly, for those who don't know PhenoArch, is a phenotyping platform that aims at analyzing the genetic determinism of plant responses to environmental conditions, mainly drought, temperature, and light, and is an automatic system for having RGB images and control of plant respiration, with a capacity of 17,000 plants that is compatible with genetic studies, genetic association studies. So the first thing we need is the incident light. While this is quite obvious when you go to the field that you have no problems for estimating this incident light, when you go to growth chambers or greenhouses, we see that there's a large spatial availability of light, because here we see the output of 8 PAR sensors in the greenhouse, yielding a significant variation in available light. And this is explained because of the number of structures, lamps, shades, columns, beams, and also the surrounding and buildings and even trees that are blocking this light, and they produce a variability, a spatial and temporal variability that can go up to 30%. One of the solutions will be installing hundreds of PAR sensors, but our technicians will not be very happy because this will be difficult to maintain and to calibrate. So what we have done is to develop a method for mapping this available light for every day of the year. Here we have our greenhouse. So what we did, we took hemispherical images of the roof of the greenhouse every square meter, and then we analyzed those images for extracting the part of these images that are related to the structures and what was the sky. And we used these hemispherical images for two things. First, we calculated the amount of diffuse light that is transmitted, and we used a standard over sky model that takes into account the zenith angle and the gap fractions. And then we interpolated these results to the rest of the greenhouse, and we obtained this kind of light map that is showing how diffuse light is transmitted in the different positions of the greenhouse. We did similarly the same for the direct light transmission, but here the things are slightly different, because this transmission of direct light, it depends on the hour and the day of the year. So what we did was to simulate for every hour and day of the year the sun, the relative position of the sun, and we calculated how this direct light was transmitted at each position, and then we interpolated to the different positions in the greenhouse, but in this case we obtained a map for every day of the year. So putting all together all this information, we mixed these direct light transmission maps and the diffusion light map, and then we obtained a local amount for each XY position in the greenhouse, with differences that were accounted between 10 and 30% depending on the season. And of course, these results were validated with the PAR sensors that were placed at fixed positions in the greenhouse. The second thing was to to create a method for measuring how much of this incident light was intercepted. And here we had a problem because compared with field experiment, where you have a plot of the same genotype, an homogeneous plot, I would say that in 99% of platform experiments, we have experiments that harbor composite canopies. That means that one plant is surrounded by plants of different genotypes. So we end up with this situation where we have a small plant with neighbors that are bigger or smaller with different areas and different architecture. To solve this problem, we used the RATP functional structural plant model that takes into account these problems by describing the geometry of the canopy with an array of 3D cells, as you can see in this image. And each cell, it may contain leaves of different plants or different leaves of the same plant. And each voxel is characterized by the leaf angle distribution inside this voxel and the leaf area density that can be derived from the 3D representations. So the first thing we needed was to obtain these leaf angles. Every day we take images, different site images at different angles for each plant. And you can see here that this image is showing a lot of information. So we see a lot of leaves and which are the angles. On the contrary, this image is not showing too much information. So for choosing which was the best image for each plant, we use the top image doing a major axis regression. And then we selected for each plant the best site image. And that image was segmented. We extracted the plant skeleton. We divided this skeleton in elementary segments. And then we compute which was the mean angle for this plant. And this method proved useful for comparing maze lines. In that case, we are comparing a floppy leaf maze line that shows an average leaf angle of about 42 degrees, compared with this one with erect leaves that shows an average leaf angle over time about 55 degrees. And what is interesting is that these leaf angles were reproducible between experiments with a high heritability. The second thing was to obtain the leaf area density from 3D representations. I'm not going to detail too much how we did this. We used, like the presentation before, a space carving algorithm that uses 12 different multi-perspective imaging, in that case 12 images, and by successive steps we end up with a 3D representation with a good length and volume resolution. So we have this 3D representation and the leaf angle distribution for each voxel. We can scale up this to the whole greenhouse. It's difficult to represent, but here you have 1,680 plants, and each of the volumes represent the voxel with the size that is proportional to the leaf area inside the voxel, and the colors represent the the dominant leaf angle class that they were limited to 5 to avoid a lot of colors. And then once we have this representation of the whole greenhouse, we feed the model with this information and with the local available light, and then we obtain the amount of light that was intercepted for each plant every day of the year. And for showing how the model proved for capturing, the model captured the effect of architectural characteristics of the plant and the competition between plants. If you compare this, the red with the green one, we see that over time this plant is competing for light with other ones, and this results with a lower amount of intercepted light. So now we have how much incident light arrives to each plant, how much of this light is intercepted, and finally we want to calculate how efficiently this intercepted light is converted to a biomass. So it's just calculated as the relation, as a slope of the biomass accumulation to intercepted light, provided that we can estimate the biovolume that is related with biomass non-destructively and quite easily. And what we have seen is that here you have 3 different maze lines growing in 2 different experiments, that they were quite similar in terms of temperature, but slightly different in terms of light availability. And what we have seen that they display different growth rates at the same thermal time. When we express these results by the amount of the intercepted light, what we saw that these rates that is the radiation use efficiency, were common between experiments. And what was interesting is that the ranking of genotypes was maintained between experiments showing a no G/E interaction. Further, we wanted to test whether if these radiation use efficiency values were related with gas exchange measurements performed in single diffs. And this was done in just 8 genotypes under 2 water scenarios, and we see that these extracted radiation use efficiency values from the platform correlated quite well with the photosynthesis and stomatal conductance. And this suggests that we can use these values as a surrogate of canopy photosynthesis for exploring genetic variability at high throughput in large collections of genotypes. Beyond the results, I'm going to present the work of Xu Chen, that he aimed at estimating other traits related with light interception, like interception efficiency and extinction coefficients. Here we have another problem, is that estimating those traits require that the plants are organized in microcanopies, sharing the same genotype. And this is not compatible with genetic analysis studies, at least with the number of plants available in our platform. So it would require at least 10,000 plants. So what Xu did was to use the pipeline that I showed before, and he extracted information for every single genotype, and he built a virtual canopy for each genotype. So in that case, he extracted all the information of the genotype B73, and he built a virtual canopy that was only of this genotype. And then he used the pipeline for extracting all the light-related parameters for each of the genotypes. And then he could extract different traits like maximum radiation interception efficiency, the leaf area index, and also the light extinction coefficient. And he extended the analysis to a panel of 250 maize hybrids, showing a large variability on all of these studied traits. Then Ott and Santiago performed a GWAS analysis on those extracted traits, and they found significant QTL for all of them. And what is interesting is that some of these QTLs co-localize with the QTLs detected for yield components in a network of experiments in the field with the same maize hybrids. And for concluding, I hope I showed how important it is to characterize spatial and temporal the environmental variability in the greenhouse, because we are in a semi-controlled environment. How these light-related parameters can be estimated using at high throughput by combining the 3D reconstructions and a functional structural plan model. And how the suite of methods that I presented here permitted the dissection of biomass accumulation in different traits having each a genetic variability. So we propose that these genetic values extracting from a phenotyping platform can be used for crop models and for field studies, opening up the way for large-scale genetic analysis of the components of plant performance. And just to thank you all the people that make possible to make experiments in the platform and obtain all this data. Thank you.